Hello, class, and welcome to chapter 11. This chapter will be on a really good introduction to the nervous system, and it's divided into three parts, and you can see those three parts in Canvas. Part A is a good review of anatomy, and I'm not going to be going over Part A in this lecture, but you are still responsible for all the information in Part A because it is a review from anatomy, and I could ask you questions on it. So for example, you should know the three overlapping functions of the nervous system. You should know how it's divided anatomically. You should know the difference between the sensory and the motor division and what makes up each. And a good chart that describes kind of the division and organization of the nervous system is this one. You should also review all the neuroglial cells, which are all the supporting cells, which are these four main groups that are seen there. So again, part A, you should review on your own. You're responsible for this information. But I'm going to have you guys review that part on your own. And in this lecture, we'll go over parts B and C. So part B, we're going to talk a little bit more about the fundamentals of the nervous system. We'll start with a little bit talk about membrane potential and resting membrane potential. So like all cells, we talked about your muscle cells, neurons also have a resting membrane potential. And unlike most other cells, neurons can rapidly change their resting membrane potential. And this is important because this makes your nervous tissue, your neurons, highly excitable, which just means they have the ability to send and receive stimuli really easily. So the basic principles of electricity, because we're talking about a charge that exists across the membrane of the neuron, are that opposite charges are attracted to each other. Energy is required to keep opposite charges separated, and energy is liberated when the charges move toward one another. When opposite charges are separated, the system has potential energy. So we need to understand what voltage is. Voltage is any measure of potential energy generated by a separated charge. In the nervous tissue in your neurons, this exists as millivolts, and it's measured between 2 points. And this is called the potential difference or potential. And again, this is the charge difference across the plasma membrane, resulting in potential. And the greater the charge difference between points, the higher the voltage. Current is a flow of electrical charge or ions between 2 points, and this can be used to do work. The flow is dependent on voltage and resistance. Resistance is a hindrance to the charge flow, and we can have an insulator, which is a substance with high electrical resistance, or a conductor, a substance with low electrical resistance. We talk a little bit about Ohm's law, which gives the relationship of voltage, currents, and resistance. And this just tells us that the current is equal to the voltage divided by the resistance. So this just means that current is directly proportional to voltage. So if voltage increases or the potential difference, the greater the current. No net current flow between points with the same potential, and current is inversely reported to resistance, proportional to resistance. So the greater resistance, the smaller the current. The role of membrane ion channels is incredibly important in the neurons. These are large proteins that serve as selective membrane ion channels. Potassium channels only allow potassium to pass through, and sodium channels only allow sodium to pass through. There's two main types of ion channels. They can be leakage channels, which are always open, they're non-gated, or we have gated channels in which part of the protein changes shape to open or close the channel. And there's three main gated channels: chemically gated, voltage gated, or mechanically gated. Chemically gated, often sometimes called ligand gated, is open only with the binding of a specific chemical like a neurotransmitter. Voltage gated channels open and close in response to changes in the membrane potential, and mechanically gated channels open and close in response to a physical deformation of receptors, such as in your receptory receptors. So here's just a look at the operation of gated channels, chemically gated ion channels, voltage gated ion channels. When these gated channels are open, the ions will diffuse quickly along their chemical concentration gradient from a high to low concentration, as well as along an electrical gradient toward the opposite electrical charge. The electrochemical gradient is then what we call the electrical and chemical gradients combined, and the ion flow creates an electrical current and the voltage changes across the membrane. And this is expressed by a rearrangement of Ohm's law where the voltage is equal to current over resistance. A voltmeter can measure the potential or charge difference across the membrane of a resting cell, and the resting membrane potential of a resting neuron is approximately negative 70 millivolts. The membrane is said to be polarized when this exists. Potential is generated by differences in ionic composition of the intra and extracellular fluids, so the interior and exterior of the cell, and also the differences in the plasma membrane permeability. So this is just a voltmeter measuring the electrical charge or the resting membrane potential that exists between the inside and outside of a neuron, specifically the axon. And you can see here resting membrane potential is about negative 70. Differences in ionic composition, the extracellular fluid usually has a higher concentration of sodium ions, and the intracellular fluid usually has a higher concentration of potassium ions. And potassium ions is what will play the most important role in membrane potential. And here is a great animation that you can watch about the resting membrane potential as well. So here's just a look at some figures that describe more about the resting membrane potential. So generating a resting membrane potential depends on, again, the differences in sodium and potassium concentrations inside and outside of the cells, and the differences in the permeability of the plasma membrane to these ions. So again, the sodium concentration is usually higher outside of the cell, and the potassium concentration is higher inside of the cell. The sodium potassium pump, what is shown here, will maintain these concentration gradients of sodium and potassium across the membrane. The differences in plasma membrane permeability, they are impermeable to large anionic proteins. They're slightly permeable to sodium through leakage channels. Sodium will diffuse into the cell down its concentration gradient, and it's 25 times more permeable to potassium than sodium, meaning potassium diffuses out of the cell down its concentration gradient. The plasma membrane is also very permeable to chloride ions. So more potassium ions will diffuse out than sodium diffuses in. So as a result, the inside of the cell becomes more negative, and this is what establishes that negative 70 millivolts resting membrane potential. And the sodium potassium pump stabilizes that resting membrane potential by maintaining the concentration gradients for sodium and potassium. And what that sodium potassium pump does is it pumps 3 sodium out of the cell while 2 potassium are pumped back in. And here's just a look at that sodium potassium pump and just the direction of sodium and potassium ions flowing through their channels. Membrane potential changes when the concentrations of ions across the membranes change and the membrane permeability to these ions change. The changes produce 2 types of signals: a graded potential, where incoming signals operate over very short distances, or an action potential, which is what we'll focus on in the nervous system, which is a long distance signal of an axon. And these changes in membrane potential are used as signals to receive, integrate, and send information. We have a couple terms to describe the membrane potential, talking about changing relative to the resting membrane potential. Depolarization is any decrease in membrane potential, which means it will move toward 0 and above, so it will become less negative. The probability of producing an impulse will increase when this happens. Hyperpolarization means an increase in membrane potential away from 0. So the inside of the membrane becomes more negative, and the probability of producing an impulse along that axon decreases. So here's just a look. Depolarization, the inside of the cell becomes less negative, and hyperpolarization, the inside of the cell or axon becomes more negative. A graded potential is a short-lived localized change in membrane potential. The stronger the stimulus, the more voltage changes and the farther the current flows. It's triggered by a stimulus that opens gated ion channels, which will result in depolarization or sometimes hyperpolarization. And it's always named according to where it's located and its function. So here's just to look at a graded potential. It'll occur at a stimulus and then it'll decay as it spreads out. Once the gated ion channel opens in a graded potential, depolarization will spread from one area of the membrane to another. As you can see this here, the spread of depolarization will occur in both directions. The graded potentials are signals only over short distances, so the current will flow but dissipate quickly and decay. And you can see that here, how the membrane potential goes back to normal after time, after the initial site of depolarization. An action potential is the principal way that neurons send signals. So we'll talk a lot about action potentials, and you just have to remember that an action potential is just the name for a neuron sending a signal. It's the means of long distance neuronal communication, and it occurs only in muscle cells and axons of neurons. There's a brief reversal of membrane potential with a change in voltage of about 100 millivolts, and action potentials do not decay over distance as graded potentials do. And in neurons, we refer to action potentials as a nerve impulse, which involves the opening of a specific voltage-gated channel. So here's a look at an action potential and what happens here. When it's closed, when it's opened, sodium rushes in. It's inactivated again when it's blocked. The voltage-gated potassium channel has one gate and two states, either closed or opened, and when it's opened, potassium rushes out. There's four main steps of an action potential, which you should know. The resting state is when all sodium and potassium channels are closed. Each sodium channel has two voltage-sensitive gates, an activation or an inactivation gates, and each potassium channel has just one voltage-sensitive gate, which is closed at rest and opens slowly with depolarization. So the first state of an action potential is the resting state, where all gated sodium and potassium channels are closed, as you can see here. And you can see here that sodium is more concentrated outside of the cell and potassium is more concentrated inside of the cell. And this graph shows you that there's no change in membrane voltage yet because everything is at resting. So when we get a stimulus, we get depolarization, which is when sodium channels open. This allows sodium to rush into the cell. As a result, the intracellular fluid becomes less negative because sodium ions are positive and they're coming into the cell. At threshold, when we get to about negative 55 to 50 millivolts, we get a positive feedback mechanism that causes all the sodium channels to open, which results in a large action potential spike where the membrane polarity jumps to about positive 30 millivolts. And that is shown here with depolarization. Sodium channels open, allowing sodium to enter the cell. And this is why we get a more positive upswing in our graph, because sodium is positive and it brings a positive charge to the inside of the cell. Repolarization then is when sodium channels are inactivating and potassium channels are opening. So potassium will now leave the cell, leading to repolarization, where the membrane returns to resting membrane potential. And that can be seen here as positively charged potassium ions leave in green. The inside of the cell will become more negative again. Hyperpolarization is when some sodium channels remain open and sodium channels reset. So this allows kind of some extra positively charged potassium channels to leave, which allows the inside of the membrane to become slightly more negative, and this causes hyperpolarization, which is a slight dip below the resting voltage. And that is shown here in number 4, the hyperpolarization stage. And these are all the stages at once that you guys can pause and look on your own. And this is a great video of how an action potential is generated. All right, resting membrane potential, and this takes us through. So the threshold and the all or none phenomenon is really important. Not all depolarization events will produce an action potential. And for an axon to fire or send its message down, depolarization must reach the threshold. And the threshold is when the membrane is depolarized by about 15 to 20 millivolts. And the all or none response refers to when an action potential will either happen completely or not at all. And an action potential will happen completely if the threshold is released. Propagation allows the action potential to be transmitted from the origin down the entire axon length toward the terminals. The sodium influx through the voltage gates in one membrane area causes local currents that cause opening of sodium voltage gates in adjacent membranes. Once initiated, an action potential is self-propagating, and since sodium channels closer to the action potential are still inactivated, no new action potential is generated there. So what this means is that an action potential will only run in one forward direction, as seen here. Coding for stimulus intensity. All action potentials are alike and are independent of stimulus intensity. The central nervous system tells the difference between a weak stimulus and a strong one by the frequency of impulses. And the frequency is just the number of impulses received per second. And a higher frequency means a stronger stimulus, as shown here. So more action potential leads to a stronger stimulus. The refractory period is the time in which the neuron cannot trigger another action potential. This is when the voltage gated channels remain open, so the neuron cannot respond to another stimulus. There's two types. The absolute refractory period refers to the time from the opening of the sodium channels until the resetting of the channels. And this ensures that each action potential is an all or none event and also enforces the one-way transmission of nerve impulses. The relative refractory period follows the absolute refractory period, where most sodium channels have returned to resting state. Some potassium channels are opening, but repolarization is still occurring. So the threshold for the action potential is elevated. So only a strong stimulus could stimulate an action potential at this point. And these refractory periods are shown here. Action potentials will only occur in axons, not other cell areas. The rate of an action potential depends on two factors: the axon diameter, so a larger diameter fiber has less resistance, so it will have a faster impulse conduction, and the degree of myelination. Two types of conduction depend on the presence or absence of myelin, so more myelin will also increase the rate of the action potential velocity. Continuous conduction and saltatory conduction. Continuous refers to the slow conduction that occurs in non-myelinated axons, and saltatory conduction occurs only in myelinated axons and is about 30 times faster. The myelin sheaths insulate and prevent leakage, and the voltage-gated channels are located at the myelin sheath gaps. Only action potentials are generated in these gaps, so the electrical signal will jump from gap to gap. And that is shown here. Multiple sclerosis is an autoimmune disease that affects young adults, and it actually attacks the myelin sheaths in the central nervous system. It turns myelin into this hardened structure called scleroses, where the impulse conduction slows and eventually stops altogether. Demyelinated axons can increase sodium channels, causing cycles of relapse and remission. So your symptoms, you're kind of losing nervous system control. Treatments are drugs that modify the immune system. You're not able to prevent it, but you can maintain high blood levels of vitamin D that could reduce the risk of development. So conduction velocity, nerve fibers are classified according to their diameter, degree of myelination, and the speed of conduction. So group A fibers are the largest diameter, myelinated. So these will be the fastest transmission. Groups B and C just work our way down from intermediate to smaller. So they will be a little bit slower, and they will serve visceral organs. Impaired action potential Impulse propagation can be caused by a number of chemical and physical factors. Local anesthetics act by blocking voltage-gated sodium channels, and cold temperatures or continuous pressure can also interrupt blood circulation and delivery of oxygen to neurons. So your cold fingers will feel numb, or the foot will feel like it goes to sleep. So that's part B, which is a really probably more one of the important ones in this chapter. We're going to quickly go through part C because part C doesn't have as much that I want you to focus on. Again, you should be responsible for all of part A, review that, part B, what we just went through, and then part C, we're going to kind of skip over the important things I want you to learn in part C as well. So here we are in part C. We'll talk a little bit about the synapse, which is just the junction that mediates information transfer from neuron to neuron or an effector cell. You usually have two neurons involved in a synapse, the presynaptic neuron and the postsynaptic neuron on either the before or after of the synapse or space. And here's a look at different types of synapses. We can have an axosomatic synapse from an axon to a cell body, an axodendritic, or an axoaxonal synapse. So again, synapses are just connections between neurons, and this is a pretty neat picture showing some of those synapse connections. So here are the synaptic connections that I'll let you guys review on your own. They can either be chemical or electrical, and most are specialized for the release and reception of chemical neurotransmitters. They're composed of two parts: the axon terminal that contains the synaptic vesicles that are filled with the neurotransmitter. This probably sounds a little bit like the neuromuscular junction. The receptor region on the postsynaptic neuron. And then the two parts are filled by the fluid-filled synaptic cleft. And the electrical impulse is changed to chemical as it crosses the synapse and then back into electrical. The transmission across the synaptic cleft impulses from directly passing from one neuron to another. It's a chemical event. And again, it depends on the release, diffusion, and receptor binding of the neurotransmitter, and it ensures unidirectional communication between the neurons. Information transfer across the chemical synapses, there's 6 steps involved, which you guys should know. They're going to be shown here, and I'll go over them briefly. The action potential arrives, voltage-gated calcium channels open, calcium enters the axon terminal, which is shown here. Calcium entry causes the synaptic vesicles to release the neurotransmitter, which is seen here. The neurotransmitter diffuses across the cleft and then binds to receptors. And then the binding of the neurotransmitter to those receptors will open ion channels, creating graded potentials. And then the neurotransmitter effects will be terminated as shown here. So this gives you all the steps. It's kind of hard to see, so you can download the PowerPoint and zoom in on those steps. Synaptic delay is just the time needed for the neurotransmitter to be released, diffuse, and then bind to the receptors. And the synaptic delay will be the rate limiting step of all neural transmission, because the rate of how fast these neurotransmitters go will depend on just the time it needs for that neurotransmitter to be released. Electrical synapses are a little less common. The neurons are electrically coupled, joined by gap junctions, and they're most abundant in embryonic nervous tissue. The postsynaptic potentials, there's 2 types of potentials: excitatory or inhibitory. So here we look at postsynaptic potentials and their summation, and they can be excitatory or inhibitory, meaning kind of saying like life as a neuron is like hearing thousands of people telling you what to do, either fire, fire, or don't fire. So this is just to say that some of these potentials are inhibitory, so they'll tell the neuron to not fire, and some are excitatory, and that just allows for more greater control of this nerve symbol. And besides that, you don't need to know too much. You should just know that these 2 types of potentials can either trigger an action potential if threshold is released, and that is shown here, an excitatory postsynaptic potential, or an inhibitory synapsis and an inhibitory potential could just reduce the postsynaptic neuron's ability to produce an action potential. And that is shown here. So it's basically like driving the neuron away from the action potential threshold, kind of into a hyperpolarized state. So that would be an inhibitory. Summation, a single excitatory potential cannot induce an action potentials, but they can summate to influence the neuron. And there's two types of summations, temporal or spatial, which I don't ask you guys too many questions on the exams, but you can take a look at what that means in these couple slides. The synaptic potential, again, is just the repeated use of the synapse increases the ability of the presynaptic cell to excite the postsynaptic neuron. The potentiation can cause calcium voltage channels to open, and long-term potentiation refers to the learning and memory of these synaptic events. Presynaptic inhibition is just an inhibitory mechanism inhibited by another neuron. And these are just the comparison again of a graded potential and an action potential, kind of referring to what we learned about before, but just a good review on that. And actually these two slides are a great review on graded potentials and action potentials. And then showing the excitatory and inhibitory as well. The neurotransmitters is just the language of the nervous system. You have 50 or more different types of neurotransmitters. They can have a chemical structure, and they're also classified by their function. Acetylcholine is one of the most common ones. It was first identified. It's the best understood. It's the neurotransmitter that is released at the neuromuscular junction, and it's degraded by that enzyme acetylcholine esterase. You have other different types of neurotransmitters, catecholamines, where dopamine is one of them, norepinephrine, epinephrine, indolamines, which include serotonin, histamine, which are in important use in the brain. Amino acids make up all proteins. Therefore, it's difficult to prove which ones are neurotransmitters. But the ones that are proven neurotransmitters are glutamate, aspartate, glycine, and the GABA amino acid neurotransmitter. Peptides, different groups of neurotransmitters are shown here. Purines, gases and lipids, we have gasotransmitters, nitric oxide, carbon monoxide, hydrogen sulfide. Gas can be sometimes important because they cause some brain damage if there's too many. Endocannabinoids act as the same receptors as THC, which is the active ingredient in marijuana. They're lipid soluble, they're synthesized on demand, and they're important in learning and memory. They may be involved in neuronal development, controlling appetite, and also suppressing nausea. The functions of neurotransmitters can be identified and classified into two groups, their effects and their actions. They can have an excitatory versus inhibitory effect, these neurotransmitters, and it kind of will take you through those different groups. So I'll let you guys read through some of this on your own. The actions can be direct versus indirect. A direct action means the neurotransmitter binds directly to and opens the ion channels. And an indirect action means the neuron transmitter acts through like a second messenger, usually what we call a G protein pathway. These will have longer lasting effects similar to hormones. The neuromodulator is a chemical messenger released by the neuron that does not directly cause these excitatory or inhibitory potentials, but instead affects the strength of the synaptic transmission. So that's a neuromodulator. Channel-linked receptors are ligand-gated ion channels. Their action is immediate and brief, which is shown here. G protein-linked receptors, they're indirect, complex, slow, and often more prolonged. And a good example of a G protein-linked receptor, which we will talk a lot about later, is the muscarinic acetylcholine receptors. The mechanism is that the neurotransmitter binds to a G protein-linked receptor, and we get into this second messenger, kind of activation system like cyclic AMP, which will be important as we talk more later in the semester. Second messengers can then open or close channels, activate enzymes, phosphorylate channel proteins, and activate genes and induce protein synthesis. So G-linked receptors are really important for that as well. The G protein signaling mechanism is like the molecular relay race, because we have like this kind of handoff of the baton going through the second messenger system until we eventually get to what it is actually doing. And I'll let you guys read through this on your own. Neural integration are neurons functioning together in groups. There's billions of neurons in the central nervous system that function as a whole to make sure everything works together. The neuronal pool is the functional group of neurons that will integrate incoming information and then forward the processed information on to other destinations. The simple neuronal pool, we can have a discharge zone or a facilitated zone. And again, I do not ask you that many questions about this. Here's a simple neuronal pool. I don't ask you guys too many questions about that. Serial processing involves the input traveling along one pathway to a specific destination. Reflexes are going to be important later on in the semester as well. These are rapid automatic responses to stimuli. They occur over pathways called the reflex arc that have these 5 components, which you should know, the 5 components of the reflex arc. The receptor, which sends on a signal through the sensory neuron to the integration center, and then out through the motor neuron to affect and some sort of effector to signal a response. So you should know these 5 steps of a reflex arc, and you might see a picture like this on an exam one day. Parallel processing, input travels along several pathways at the same time. So if, for example, a sensed smell may remind one of an odor or any associated experiences. And that's why it's often said that the sense of smell is very important for bringing back memories, because they travel along these parallel pathways. Circuits are patterns of synaptic connections in neuronal pools. They can be diverging, converging, reverberating, or parallel after discharge. And again, I do not talk a lot about circuits. We'll talk a little bit about diverging, converging, reverberating, and parallel after discharge circuits when we get through different parts of the body. But otherwise, you don't have to worry about circuits for this exam. Thank you for listening to Chapter 11. Again, you should study and be responsible for Part A on your own, and then for the important aspects that I talked about in Parts B and C. And thanks for listening.